such as the Black Death. But just as we have found innovative ways to combat our bacterial enemies, they too have remained resilient and have forged pathways to survive in this medicinal war zone. This has caused the uprise of superbugs. One of the more well-known superbugs is MRSA. A superbug is a bacterial strain which is resistant to many different forms of antibacterial treatment, making a bacterial infection just as deadly today as it was in 1330. So, what do we do about this? Well, thanks to innovative minds such as those at the Detweiler Lab, there's promising research into new antibacterial therapeutics. This purple blob that you are seeing here is a macrophage, which is a white blood cell that is circulating through your bloodstream as we speak. What the Detweiler Lab has focused their research on is finding compounds which inhibit bacterial growth inside of macrophages. The compound I've been given the opportunity to work with is named 1.11. My job over the past 8 weeks has been to find the mechanism of action for 1.11. So in other words, how is 1.11 inhibiting bacterial growth? In order to do this, I will be using essential Bacillus subtilis genes knocked down with CRISPR-I to find synthetic lethality with a sublethal dose of 1.11. Okay, I know that's a mouthful. Let's break it down. So first, let's understand CRISPR-I. In order to understand CRISPR-I, we need to understand unedited normal gene function and CRISPR-edited gene function. So if we look here at the unedited gene, what we will expect is fruitful protein production. Now let's compare that to CRISPR-edited genes. As you can see here, we have these tiny molecular scissors which work to cut a gene out of the genome. Along with the tiny molecular scissors, we have this red hairpin loop, which is our single guide RNA. This guides the scissors of where to cut. If a gene is successfully spliced out of a DNA sequence, this will result in no protein production from that gene. This is known as a gene knockout. Now, if we compare this to CRISPR-I, you can see that we don't have these molecular scissors, so no cut in the DNA will be made, but we still have the presence of this single guide RNA. This will result in an interference in the DNA, hence the name CRISPR-I, CRISPR interference, resulting in subsequent protein production, but to a much lesser extent as what we see in unedited genes. This is known as a gene knockdown. So how will I utilize CRISPR-I? The bacteria I'm working with, Bacillus subtilis, has 4,100 genes in total. Out of those 4,100 genes, 258 of them are considered essential. I will be working with 258 CRISPR-I strains, each strain having a different gene that has been knocked down. So my second part to this equation is the sublethal dose of 1.11. What you are seeing here is a dose-response assay. My highest concentration of 1.11 is 75 micromolar as seen here. As we move towards the right, the concentration gradually decreases. Green color indicates no bacterial growth, and red color indicates high bacterial growth. At right about 20 micromolar, we can see a clear delineation between no growth and high growth, meaning any concentration above 20 micromolar is lethal, resulting in no cell growth, and any concentration at or below 20 micromolar results in cell survival. So, how will I use these to find synthetic lethality? You can think of synthetic lethality as combined lethality. So if we look at the left-hand side, this is my CRISPR-I bacteria, so a bacteria which has had an essential gene knocked down. If left with no further intervention, this bacteria will remain viable and healthy. Now let's look at the right-hand side. This is my bacteria which has no gene knockdowns that is treated with a sublethal dose of 1.11. This bacteria too will remain viable and healthy. What I'm looking for is when these two treatments are used in tandem, if they result in cell death. So for my test, I will treat each CRISPR-I strain with 2 treatments. The first treatment will be my control, so no drug will be added, and my second treatment is my sublethal dose of 1.11. 3 time points, 0 hour, 8 hour, and 24 hour will be taken for each treatment. If we look over at the control treatment, what we should see over the 3 time points is a steady increase in growth. Now for my treatment of 1.11, we can see one of two outcomes. The first outcome is if no synthetic lethality is present, what we will see over the 3 time points is a steady increase in growth comparable to that of the control treatment. Now, the second outcome we can have is if synthetic lethality is present, what we will see over the 3 time points is a steady decrease in growth. I will refer to this growth pattern as a hit. So this is an example of a hit I got from the CRISPR-I screen. For your reference, as we move up the y-axis here, this is the amount of bacterial colonies in the logarithmic scale, meaning as we move from 3 to 4 or from 4 to 5, there's a 10-fold increase in the amount of bacteria. Now, as we move along the x-axis, these are my 3 time points, 0 hour, 8 hour, and 24 hour. What this graph is telling us is that when a Bacillus subtilis bacteria has the MEN-A gene knocked down and is treated with a sublethal dose of 1.11, over the 3 time points, there will be a noticeable decrease in growth as compared to the control treatment. So out of my 258 strain screen, these were the 9 hits I got. So all 9 of these genes present synthetic lethality. So my next step here was to think like a scientist and try to make sense of it all. So I had my 9 genes that I knew presented synthetic lethality, but I had to figure out how do these genes work together to paint a picture of what 1.11 is doing inside of the cell. So what I did was individual research into each gene to find its function. And in one of the papers I came across, this diagram was present. I was excited to see that every single gene I had a hit for was present in this diagram. This is exciting, guys. Be excited. What this diagram depicts is menaquinone biosynthesis. So one of the main functions of menaquinone is to glycosylate membrane proteins. Again, another scary word. I'll explain. Protein glycosylation is the process of attaching a sugar molecule onto a protein. This is important because it not only determines the protein's overall structure and function, it determines the protein's overall stability. So knowing this, my hypothesis is that 1.11 is inhibiting menaquinone biosynthesis, and as a result, protein glycosylation will not happen, which will lead to membrane permeability and eventually cell death. So how will the Detweiler Lab move forward with this information? They will perform a series of tests to further validate my hypothesis that 1.11 is inhibiting menaquinone biosynthesis. If they prove this to be true, this is exciting because it proves that menaquinone biosynthesis is a targetable pathway for new antibacterial medications. So while 1.11 itself may never be used in clinical settings, it can serve as the prototype for a variety of drugs that will be, acting as a new weapon in our arsenal against superbugs. Thank you.